Okay, so this is Abiogenesis II. Now, if I recall, I was just talking about Anton van Leeuwenhoek, the cloth merchant in Holland, who used small monocular microscopes, which he made by hand. He made excellent spectacles as well. Spectacles, okay, fancy word for glasses. These were monocular microscopes and with excellent magnification and optics, 275 times or so. He discovers this unseen world, the unseen world of microbes. He's the first to see so-called animalcules, animal-like protists, amoebas, protozoans, various types, paramecia, ciliates, sarcodines, I'm sure he saw, flagellates like Euglena, and so on. Okay? That was about the late 1600s. And so this idea, the spontaneous generation of microbes, just wouldn't go away. And by the mid-1760 or so, Spallanzani, who is probably the world's greatest reproductive biologist, probably the first reproductive biologist, is convinced that microbes must have parents too, like the larger organisms that he's investigating. He's the first to show, by the way, fertilization of an egg by sperm in vitro. Now, what are in vitro? Vitro, vitreous glass. So when you hear the word in vitro, think a petri dish. Okay? In vivo means within life. Vivo, life. So he has several incredible breakthroughs. Like many Renaissance men, incidentally, his mentor and teacher was a woman. I need to mention that. And she inspired him to do scientific research. He was a scholar, a classics scholar, and also studied the law, but then turned to science. And he made these incredible discoveries in reproductive biology. He also did some insemination experiments, which is kind of interesting. I won't elaborate. Okay, so he's convinced that the microbes are floating around on dust particles, essentially, or spores or something, and they just merely fall on food and stews left out. They become inoculated, and then decay takes place as a result of bacterial activity. But how to disprove the spontaneous generation is going on. Basically, he does an experiment which is similar to rabies. Okay, so can you think of what that might look like? Two tubes here. They were small tubes known as ampules. Just think test tubes. Okay. In one, he puts a nutrient broth, like chicken soup. Okay, so chicken soup. He just leaves that one out. What do you think? Would that be the control or the experimental group? Nothing done to it? Control. Okay? The experimental group. What do you think he does here? Well, he needs to prevent the organisms from getting to it. Okay? So what he does is he seals this off by heating. So when you heat glass, it melts. And if you do it right, you're able to make a nice tapered ampule here, sealing off completely the contents. That's a hermetic seal. Okay? Now, of course, since he believes there are germs everywhere, and there really are germs everywhere, they're ubiquitous, they're all over the place, okay? They're on your skin by the billions, okay? They're everywhere, in your hair, on desks, on just everywhere, okay? They're everywhere, and in the rug, everywhere. They're all around you. You're breathing in every day, probably millions of spores of various types, okay? Good thing you have an immune system, right? You'll learn more about that later. So to make sure that there are no microbes in here, that because it could have possibly it could have been contaminated while he was sealing the broth in there. He heats it to a high temperature and boils it. Okay, killing off any of the microbes in there, reasoning that if spontaneous generation is true, it wouldn't make any difference. The microbes would spontaneously generate anyway. I mean, that's what we mean by spontaneously generate. So non-living material would give rise to living material. But he's convinced that that won't happen. And he's right. Almost every time he does this, he sees no spontaneous generation in the sealed test tube. However, there is a problem, though, because there are endospores. Unknown to Spallanzani, there are bacterial spores that are resistant to boiling. They can survive boiling. In fact, in a later video, we'll talk about organisms that actually have survived outer space, and I'm not talking about human beings. Germs that have survived on the moon for up to about 2 years. Germs are pretty incredible. Bacteria are extraordinary. Now, so once in a while, he gets an ambiguous result, and he does see spontaneous generation in here. Now, to complicate matters, there's another scientist in England doing similar experiments. His name is Needham, and he's using thick broths like mutton gravy and stuff like that. Unknown to both of them, you really have to heat mutton gravy to a much higher temperature to sterilize it. Okay, and he's getting all kinds of confounding results, and he comes away from these kinds of experiments convinced that spontaneous generation is actually occurring. And so this controversy rages on for nearly another 100 years until Pasteur comes along. Incidentally, there was an ingenious Frenchman by the name of Pasteur who knew of Spallanzani's experiments and starts the canning industry, and that's one of the reasons why Napoleon was able to take over the world, conquer the world at that time, because the French had a new technology for preserving food. There's a funny story I can tell you about that, but later ask me in class. In any case, let's go on to what Pasteur does. And I failed to mention this, but when Spallanzani seals his ampules, of course, no air. He seals them hermetically, and then he boils it. And so no air can reach the nutrient solution. He has detractors that say, oh, Spallanzani, you haven't disproven spontaneous generation because air hasn't gotten in there. Air is carrying the life principle. All right? The life principle. People are convinced at this time, of course, that life is extraordinarily different from non-life. Okay? This is before biochemistry, before organic chemistry, before our understanding of carbon compounds and carbon chemistry. They're convinced that life is life because of a vital principle, a vital force. You know, and this idea is still with us, okay? Chi. Chi. Pow! You know, karate. Right? Watch this. I'm going to break 1,000 boards with one chop. Pow! You know, he focuses his chi, his life force, right? He breaks the boards, he breaks the bricks, he does all this crazy stuff, right? Yoda. Yoda comes to mind, right? Yoda. Star Wars. Remember, Luke Skywalker is trapped on that jungle world, right? And he's going through his training with Yoda, and it's not going well. And he's convinced he's got to make it back, right, to help the resistance against Lord Vader. and he doesn't know what he's going to do. His spaceship is in the lagoon, and of course Yoda comes on, and he raises the spacecraft out of the lagoon, right? You remember that scene? That's great stuff, okay? Sets it quietly down, right? And Luke is just astounded, right? The Force. May the Force be with you. The Force is everywhere. This is the idea of vitalism. There's some kind of spooky, supernatural force around us, okay? And it imbues all of life. It's what makes life different from non-life, okay? Vitalism. Is it true? How is Louis Pasteur going to disprove this? This is how he does it. Okay? Comes up with this really pretty novel idea. He creates a flask with a swan neck. Okay? There we go. So here's the vessel. It's open at one end. All right. He puts in the chicken broth or the nutrient infusion. Okay. He inoculates it. Over time, the microbes grow, become turbid, just as in Spallanzani's experiment. You could see the turbidity as the number of microbes increase. Billions of them will be there in a few days, or actually in less time than that, multiplying exponentially. All right, so it's turbid. You know the germs are in there. You can even sample it if you have a long wire with a hook through there, and then check it with a microscope. All right. Now, notice that it's open to the air, so the vital principle can get in, right? Now, he boils it, just as Spallanzani did, okay? Killing all the microbes, everything dead, all right? Over time, he watches this. No spontaneous generation takes place. Why don't the microbes ever get in there? Well, it's simple. They get trapped here in this gooseneck of a flask. They get deposited here, so they never make it into the nutrient broth. No spontaneous generation. Air can get in there. If it's carrying the vital principle, then why doesn't the vital principle then produce spontaneous generation of microbes? Because there is no vital principle. Louis disproves. Louis Pasteur disproves vitalism. And he disproves the spontaneous generation of microbes. Now, he does a control, and he does many other experiments like this, and the scientific community is convinced that a spontaneous generation of microbes is impossible. So now we've got a huge quandary on our hands. Because by this time, it's fairly clear that life comes only from life, biogenesis. If that's true, and I say this is the first life, then you say, oh, wait a minute. There must have been life before it then. Well, yeah, that's right, biogenesis. And if that's the first life, then of course there must have been life before this. So what do we have? An infinite regress, going all the way back to infinity. That doesn't make any sense. You know, just doesn't make any sense. Clearly that can't be the case. So where does life come from? Again, we're left with this huge enigma. And it isn't until the 1920s when Oprah and Haldane together, actually separately, Oprah in Russia, a plant physiologist and biochemist, and Haldane, a zoologist, come up with the heterotrophic theory of the origin of life. I have to stop it now.